Great. Hopefully everybody can hear me pretty well on the Zoom there. It seems that I have to lean forward a bit to make sure that the mic picks up my voice. I guess let us know in the chat if you're having trouble hearing over Zoom. I'm Ty Roachford. Welcome to my dissertation proposal talk, where I'll be talking to you about the functional implications of co-transmission. Co-transmission is when one brain cell sends two different types of chemical messages. And so we'll get into that and what it means for neuroscience. But before we do so, we'll start off with how we study the brain in general. So the brain is one of the most complex objects in the universe. Maybe people say besides the universe. And so if you want to understand the brain, then you understand the brain like we attempt understanding the universe by breaking it up into its atoms. And so the idea is, what are the atoms of the actual brain? Well, it's the brain cells. And in particular, when it comes to brain cells, we're very interested in the voltage of these brain cells. You see, brain cells are electrically charged. They're more negative inside than they are outside. And when we take an electrode and actually stick it inside the neuron to measure this voltage, we can see that the voltage fluctuates. It doesn't just stay constant. And there's a particular type of voltage fluctuation, which I have pictured here, that you'd call an action potential. And an action potential is important in the brain because it signifies the release of chemical messengers or neurotransmitters, which are important for how the brain cells actually communicate with each other and able to brain to do all of the wonderful things that it does. The problem is there are a ton of different neurons, 80 billion in the human brain, more than that, thousands of different types. And so we need a way to actually understand this diversity and complexity that we see, because we can't just do it on paper or understand it sheerly with equations. And the solution that we have here is computational neuroscience, and that's the direction that I'm taking in this dissertation proposal. What we do is we acquire experimental data, however that is. It could be recording cells like I talked about earlier, or it could be actually taking these brains, slicing them up into little tiny slices, putting them into a computer, reconstructing them to get their precise digital shape, and then actually simulating the dynamics of those neurons with that particular shape so that we can understand all the different sorts of neurons that we actually see in biology. And so this is a good way to actually understand the behavior of thousands and thousands of neurons in a way that we couldn't possibly do without computational neuroscience or analytical computational methods. And this results in a kind of virtuous cycle between experimental and computational neuroscience, where they each inform and help each other. So what happens is that we take the data and we construct models. We run simulations of those models to understand what it is that the actual thing we're modeling might be doing. And then we use that to inform experiments, which then we collect more data from and begin the cycle all over again. So getting into sort of the data that we've collected about neural transmission and co-transmission in general, not being super duper exhaustive here, we have two different neurons. We have the before cell and the after cell, also known as the pre-cell and the post-cell. And the situation here is that we have chemicals going inside the neuron. They go in through the dendrites, they change the voltage in the cell body. This eventually sends a signal known as the action potential down the axon segment to the presynapse, where neurotransmitters are then released onto the next neuron, which starts the cycle all over again. This is typical neural transmission and what we like to call monotransmission, as opposed to co-transmission, where the chemicals, the neurotransmitters that are released into the presynaptic cleft, are only of one particular type. We have this diagram here just indicating that eventually when the voltage reaches a certain point, then that's when you get this crazy voltage spike that is characteristic of neurons. So co-transmission, which is a novel mode of neural transmission that we're just finding out, where the neuron releases two different types of chemical messengers, is being explored as something that seems to have some really, really cool things to imply about how brains actually work and solve the problems that they do solve on a daily basis. Right here, we're zooming into a synaptic cleft where we have the axon terminals, the presynapses, releasing chemicals into the cleft and the dendrites on the other side which receive them. And so these signals can either be excitatory, where it raises the voltage of the postsynaptic neuron, and we can see that here. This is excitatory monotransmission, where the particular chemical that we're looking at in this case is called glutamate, and the receptor for it is called AMPAR, or AMPA receptor. And so it causes this positive deflection, or what we call an excitatory postsynaptic response in this postsynaptic neuron. And then we also have inhibitory monotransmission, representing with these red dots, GABA, which binds to a receptor, the GABA receptor, and causes a hyperpolarization or more negative voltage potential of the neuron. And so the cool thing that we're seeing with co-transmission is that we get a biphasic signal that looks like this inside of the postsynaptic neuron, where you can have inhibitory before excitatory effects on the postsynaptic neuron, but then you could also have excitatory before inhibitory, depending on the actual timing release and time constants involved in this signal here. And so while we're talking about co-transmission generally, which is one neuron releasing two different neurotransmitters, there's also the case of co-release, where we're releasing two different transmitters, but not through two completely different presynapses, but from the very same presynapse. And not only that, but from the very same vesicle, where we have the vesicle actually being loaded up with both glutamate and GABA. Okay. So co-transmission has also recently been found to be widespread throughout the mouse brain. This was a study done by one of the postdocs in my lab, Cesar Ceballos, along with his team at the time, and they saw that inside the mouse brain, 7 out of 12 of the regions that they surveyed had this sort of reverse, let's call it, biphasic signal, and the 9 out of 12 of the brain regions had this straight signal. And so the straight signal is where you have inhibition, then excitation, whereas the reverse signal is where you have excitation, then inhibition. And this just results in a biphasic signal that looks a little bit different, being flipped about the x-axis. So this begs the question, if we're seeing it in so many different brain regions, both versions of them in so many different brain regions, why is co-transmission so widespread? Why should it be everywhere? We were understanding brains with monotransmission pretty nicely, so it seemed so. What's going on? Well, co-transmission may support unique functionality that is impossible with monotransmission alone. Perhaps that's why we're actually seeing it pop up in these places, and perhaps in places where we've modeled brain areas but didn't quite get the right results could be because we were ignoring co-transmission. And co-transmission may allow for more energy efficient processing than monotransmission. So although it is more metabolically costly for co-transmission to occur in a single neuron because of the extra molecular machinery required to pack those neurotransmitters into the vesicles, it's still going to be less expensive than having two completely different neurons that have to maintain their own metabolic costs and have two different sorts of neurotransmitters. And so it could be both of these things, that it's not only more energy efficient and provides unique functionality, but it could also be neither of these things, in which case we definitely want to know why is it so widespread, because it doesn't seem to be something that should be the case. In nature, we often experience that everything has just enough of what it needs to kind of get by. And so being that it's metabolically expensive, it doesn't make sense for it to sort of show up and have no purpose at all. So another reason to look into co-transmission is that failures in co-transmission have been associated with numerous sorts of diseases and disorders, depression, alcohol intake, Parkinson's, Alzheimer's, epilepsy, most likely many, many more. It's hard to get a good say of this because it is a bit difficult to investigate co-transmission, but we've come up with some awesome methods, and computational methods certainly help investigate this as well. Yeah. So this is the plan of attack. We take all of this data that we've collected, we put it inside of models. We take the models, simulate the behavior, inform our experiments, and collect more data to do the same thing again. And so how do we actually model these neurons? How are we doing it? Well, we're modeling them as electrical circuits. It turns out that these things are a perfect match, electrical circuits. They call them equivalent circuit diagrams, where you have the capacitance. Let's start from here because you have the cell, and the cell has more negative particles inside than outside, and that causes what we call the membrane potential. And so this actually ends up acting like a capacitor because of the separation of charges. And the actual channels, ion channels that are embedded inside of this membrane, this bilipid membrane layer, act as resistors, which bring ions inside and outside of the cell at different levels of conductances. In other words, the higher the resistance, the less ions that it will let inside or outside of the cell. And conductance is the inverse of resistance. And so you could also talk about it in these terms. Suffice to say that modeling neurons as a RC circuit, a resistance capacitance circuit, is the go-to method in the literature. And so that is what we're doing here as well. And then just to say that this little IT input current here can be sort of an external current that we're driving with an electrode, but it could also be sort of synaptic currents that are coming from other neurons. On the side here, we just have showing that in the case of an external current, we end up having this sort of shape in the current plot versus time. And the response to that in terms of the voltage after you inject this current looks sort of like this. Okay. So getting into how this is actually applied when it comes to neurons, because we have to sort of understand the variables now in terms of how they describe the neuron. Well, we have the capacitance here, we have the voltage here, and we have the current terms on this side. And so what this is describing is a Hodgkin-Huxley neuron, and it's describing the change in voltage over time according to that particular neuron's properties. So we have the conductance L, and this is standing for the leaky conductance. And this is just kind of describing the ion pumps as well as maybe some of the channels that we're ignoring modeling very specifically, but allows us to capture the behavior of the Hodgkin-Huxley neuron or the neuron even still without going into super duper detail. In other words, without actually defining all of the possible ionic conductances and synaptic conductances that this neuron could possibly have. We can keep it simpler for the sake of just modeling. And so what we're using here is these ionic currents for a sodium persistent channel and a hyperpolarization activated channel. And on this other side with the synaptic currents, we are modeling AMPA and GABA respectively. So excitatory and inhibitory types of neurotransmitting signals. It's particularly interesting to look at these because they are opposite in their effects. And so we expect to see interesting stuff as we'll see coming. Okay. So it turns out in our preliminary studies that co-transmission induces postsynaptic filtering properties. At least this is what we've seen, where they sort of model low pass, band pass, and high pass filters, where when you have a postsynaptic neuron, or a neuron that is receiving co-transmission, then essentially, depending on what are called the rise times of the signal, then we can get varying different sorts of postsynaptic summation patterns, where we have an inhibitory pattern, simply put, which kind of models low pass. We have transient, which is modeling band pass, and excitatory, which is modeling high pass. And the idea here is that when you have a low pass filter, it does not let through any filters that are any signals that are too fast for its own particular rate. And the same thing, but opposite, for when we're talking about high pass. In high pass situations, we don't have any signals that are slower than a certain threshold being let through. Whereas with band pass, we can have very particular sort of frequencies that are being responded to in between. So the rise times here, you can think of the rise time that we're varying as the sort of slope right here. So if we were to consider this as being an actual current, then the way that or conductance, then we can see that a slope that is more vertical, more towards vertical, more slanted, then we have a faster time constant. The response to whatever voltage that goes upon the postsynaptic cell is going to be quickly nullified or taken out. So it bumps up the voltage a little bit excitatory, and then it quickly comes back to its resting potential. Maybe it bumps it a little bit inhibitory, and then it quickly, again, comes back to its resting potential. And so this is describing the rise times and the decay times. It is the degree of slope, the amount of slope that the current actually has. Okay. We investigated the conductances too, not just the rise times. And again, let me just be clear that you have a postsynaptic neuron and it can get a signal that is a strong signal. It'll take the voltage up from, say, -70 to -60. And the speed at which it takes you from -70 to -60 is what we call the rise time. If it's very, very quick, then we have a small rise time. And if it's very, very long, it takes forever to get from -70 to -60, then we have a very long, large rise time. And this, as well as the conductance, the strength of that signal I was previously talking about, is what actually influences whether you get these sorts of postsynaptic summation patterns the most. We tried looking at latency, the difference between when these two signals actually arrive at the postsynaptic cell, but this did not have as much influence on inducing these sorts of postsynaptic filtering patterns that we see here when we varied both the rise time and the conductances. The thing is, yeah. So then we took co-transmission and we wanted to compare it to monotransmission to see, okay, we can see that there are these interesting baseline patterns, postsynaptic summation patterns that get induced in the postsynaptic neuron. But what about when we have monotransmission? Can monotransmission do the same thing? Well, in realistic scenarios, you don't have the case where you have a presynaptic neuron, like in this case, you have two presynaptic neurons that are monotransmitting, both transmitting onto the soma. Typically, what you have is that the excitatory monotransmitting cells will release their signal onto the dendrites rather than the soma, whereas the monotransmitting inhibitory cells will actually release their signal onto the soma. And so to match that, to actually, fairly compare in a biologically realistic way, monotransmission versus somatic transmission, we compared those signals across those various rise times as seen before. So we still have over here, the low pass, band pass, high pass, or inhibitory, transient, and excitatory, if you want to say it like this, type summation patterns. But in the case of dendritic co-transmission, we did not have this. We only had these simple excitatory summation patterns, which provides evidence that, okay, maybe monotransmission really can't do the same job that co-transmission can do. Not shown is that we also had this sort of dendritic coupling constant between the dendrites and the soma, which determines how much the voltage change in the dendrites changes the voltage in the soma. And essentially, not until we had a very high number of this dendritic coupling constant, such that as soon as you change the voltage by a few millivolts, then the soma also changes by a few millivolts, did we have the same sort of behavior as somatic co-transmission. So just more preliminary evidence for why monotransmission may not be able to do the same job as co-transmission. Moving on to modeling co-transmission at the network level. We've got preliminaries in understanding how exactly the constraints of monotransmission fail to allow it to have those same sorts of unique postsynaptic summation patterns as we have in the monotransmitting case or in the co-transmitting case. And so we want to understand better how these things actually show up at the network level. And so for this, in large simulations, large networks, we use leaky integrate and fire neurons, which is a reduction in complexity of the neurons that we used before, the Hodgkin-Huxley neurons. In this case, we're not worrying about very specific sodium channels like sodium and the HCN or hyperpolarizing activating channel. We're just worried about excitation, inhibition, as well as excitation, inhibition, as well as this, this just like this leaky current that is going on here. Okay. And so this reduction in complexity, all we have is the decay times. We don't have the rise times, and that's because we sufficiently explored rise times. And so we want to get a better idea of what decay times can actually do as far as influencing network behavior. So to investigate co-transmission in large networks, what we've done here is we have two populations of neurons, excitatory neurons and inhibitory neurons. And both of these populations are getting inputs from an external excitatory input, as we can see for both the monotransmitting and the co-transmitting network. And so in the case of the co-transmitting network, we take the inhibitory population and we actually make this the co-transmitting population, so that instead of just releasing inhibition or GABA, then it's going to also release excitation or glutamate. It's a proxy for this. Even though we're not being as specific as the Hodgkin-Huxley neurons, we can still get results that help us understand what it is we are studying without being super detailed, which is very computationally expensive. So co-transmission in large networks with these leaky integrate and fire neurons allow us to do what is computationally intractable when we approach it with such detail. And as a result, we've seen that co-transmission induces control over various network level properties. So we have a bunch of different network level metrics that we can measure, and these are plots of three of them. We have the correlation of variation of interspike intervals, and essentially this is just telling you how when the neuron sends its signal and there's time between each signal, how is the time between each signal related to the time between other signals? How correlated are they? If we have a very high correlation, then we have bursting, meaning it spikes a lot and then it maybe stops for a while and then it spikes some more and then stops for a while. Whereas if we have more towards one, that means it's evenly spaced. All of them are correlated about the same. And so we can expect regular firing. Whereas we next now have the firing rate, and this is just generally how much are the neurons firing at all. And so we can see that blue is going to be the smaller value, so smaller correlation of variation of interspike intervals. Firing rate, blue being smaller amount of neurons firing on average. And then we have EE synchrony, excitatory synchrony, where we're actually seeing, okay, how often do these excitatory neurons fire at the same time? How often are they actually synchronized? And we did this for just a sample of them. And we can see that with various different time decays for all of these, we get these different various results in both monotransmission and co-transmission. And so rather than getting super duper hung up on the details of each one of these metrics and what they mean, let me pop over to this and show just a few more, where we have the binary entropy, analog entropy, binary energy efficiency, which is showing on a high pass what does the information capacity, the binary entropy is like the information capacity of the neural network when considering a binary code, when it's not about considering all of the spikes of the neuron, but rather than just did a whole large population spike or not. If we consider it like this as a code where we're considering sort of, did they fire above a certain threshold to make this signal happen? We're relating that now, this information capacity in this binary mode, to the decay times. And in this case, we're doing it with analog, where we're counting actually all of the spikes. We're not doing sort of any thresholding here. We're worried about the actual rate and the amount of information that can convey, given that a neuron spikes at its a time that is slower than its fastest possible spiking time. And similarly here, the binary energy efficiency is actually telling us, okay, how efficient is this binary code that we're considering? And we have an analog entropy binary efficiency too, but it did not make it here into the slides. So the point now, again, to just get out from all of the details, because these are a bunch of detailed metrics, is that we can see that instead of being diagonal, like we have in the monotransmitting case, for all of these different metrics, we now have this vertical sort of orientation for these metrics across the co-transmitting case. And we believe that this suggests that co-transmission induces inhibitory control over all of these constants. In other words, all of these properties. In other words, for monotransmission, you have to modify both your inhibitory and excitatory decay time in order to end up in a particular spot where you have particular sorts of behaviors, both quantitatively and both visually and quantitatively. And in this case, the co-transmitting case, it looks like in the majority of these, all you really need to do to switch between different metric sort of profiles, paradigms, regimes is modify only your decay inhibitory time. And so it seems that this would give nature essentially an easy way for it to adapt to the situation around it by simply changing one parameter instead of two, which we expect would probably be less metabolically costly, depending on the actual mechanism. Okay. So we got the data, the models, and the simulations, and now we're going to talk about the wet lab experiments that we're going to do to be understanding these simulation results and verify that they actually happen inside of the biological organism. And if not, then we'll be able to take the details that we find about it, put it back into the simulations and models, and then use those new models to inform what to do next in terms of wet lab experiments. So what we're looking at for investigating co-transmission in the biological organism is the dentate gyrus of the mouse. And so it turns out that these CCK interneurons inside of the dentate gyrus of the mouse have been found to do GABA and glutamate co-transmission. So the dentate gyrus is very important in terms of pattern separation, which they do through the granule cells. These are the principal excitatory cells of the DG that pass on the actual signal. And so they're heavily modulated by these CCK interneurons. And we'll see how co-transmission actually helps them possibly get this done, or adds more flexibility, more possibilities for them to do computations, things that monotransmitting setup could not do. So we wanted to take a look at the properties of these cells just in simulation before we investigate them in the actual lab. And we took Hodgkin-Huxley neurons of the sort that I showed previously and physiologically parameterized them accordingly. And when we did that, we saw the same sorts of results that we saw in our last slides, where we have the same sorts of excitatory, transient, and inhibitory postsynaptic summation potentials that depend on the rise times between the excitation and inhibition. More interestingly is when we took them into a sort of dentate gyrus-like connectivity architecture called lateral inhibition. In lateral inhibition, it supports a sort of winner-take-all dynamics, and we'll illustrate that. We're illustrating that here. We well know this when it comes to monotransmitting neurons, but now we'll get to investigate and see what happens when you have co-transmitting neurons involved in the lateral inhibition. And so every neuronal cell has a target resonant frequency, a frequency that makes it fire the absolute most. And when this happens, you have this connectivity and it fires the absolute most. The CCK cell is getting activated and is inhibiting the adjacent granule cell. So when it is not getting its target resonant frequency, it effectively loses the competition and the signal that it passes on is suppressed. Whereas with the one that is winning the competition, it gets to communicate its actual signal, input signal. And so when we did this with co-transmission now, we saw something strange. We saw that even though it is getting its target resonant frequency, this granule cell, it is no longer communicating its signal, rather it is suppressed. And the one that is receiving a non-resonant frequency distractor is actually now winning the competition. And so it seems that co-transmission, at least in the small networks that we've studied here, provides flexibility by allowing the granule cells to fire in unorthodox circumstances, because we can also change what frequency that the granule cell prefers by modifying either the conductances or the rise times that we saw earlier. And that will enable it to win the competition using various, different frequencies. Interestingly, another thing that has happened with these CCK plasticity cells is that environmental enrichment shapes the way that they behave. And so early development robustly increases, or environmental enrichment during early development robustly increases depolarization-induced suppression of inhibition, along with cannabinoid punctas, glutamate puncta, and GABA puncta, indicating that, these things are strongly remodeled by experience. In other words, this is going to give us an opportunity to be able to explore these networks of CCK and granule cells at different levels of inhibition and glutamate, and also different levels of depolarization-induced suppression of inhibition. And so what DSI is when the postsynaptic neuron that's being depolarized actually releases cannabinoids onto the presynaptic neuron, thereby lowering the amount of GABA that it actually releases. And this kind of, like, in some sense, mimics altering one's GABA decay times. We hypothesize that co-transmission levels are linked to the filtering property that modulates input and output selection and differentiation. In other words, with this ability to actually change your own rise times, decay times in order to have different winner-take-all dynamics, you could possibly store more patterns and therefore be a more effective dentate gyrus doing your job. And co-transmission and modulation of the inhibitory time constants, but also other time constants, may be what allows for this to happen. And this is something that we'll investigate too, investigating both the mouse in the standard environment and the enriched environment to understand how these details actually modify the circuit at a biological level. So the procedure with those things set up now that we can actually target the very specific cells that we need to target genetically, we're using optogenetics. And so we take these CCK cells that we were able to target, we give them rhodopsin so that when we shine light on them, we can cause them to fire at various different rates. And then we can actually take an electrode and measure the granule cells to see what the postsynaptic potentials, currents, voltage actually ends up looking like. And so once we have this set up here, we are going to use DNQX to block the AMPA receptors or glutamate signaling. We can use gabazine to block the GABA A receptors, GABA signaling, and we'll analyze how the shape, time, and summation of the synaptic inputs change. We'll be able to see what it is different here than in our models. So should we introduce this DSI, depolarization-induced suppression of inhibition, and things like that to refine our computational models and produce more experiments. Okay. And so this is the virtuous cycle. We take the data, we put it into models, simulations, experiments, and go on so forth again. We expect that we'll get results that are very similar to our simulations. We'll probably expect to see that we'll get the filtering sorts of properties, and that these properties will be able to be shifted when considering different levels of inhibition or a different decay constants. And we'll further note whether these filtering properties vary across different synaptic locations. And this will enable us to make more, you could say, specific models to the mouse for further explorations downstream when we're doing wet lab experiments. It's important to be able to get that variability across terminals correct. And where we can go from here is looking at the same sort of stuff, but in different animals. So in the fruit fly, GABA and acetylcholine co-transmission, we think that it could help do something. So we're preliminary studies right now looking at the giant fiber neuron inside of the fruit fly, which is responsible for helping it escape to the response of a looming predator. So when there is an object or a shadow above it that's increasing in size, indicating that something might be after it, the giant fiber fires within milliseconds, allowing the fly to jump, flap its wings, and take off. And if you need to make these very precise decisions on a millisecond level timescale, then it would seem that co-transmission would be able to help for this. And we're seeing some interesting stuff here. Currently working on understanding how the giant fiber neuron is modulated by GABA and acetylcholine monotransmitting receptors. And soon we'll have the data for co-transmission inside this and be able to run simulations of this as well. We've actually simulated the entire ventral nerve cord, and we're moving towards, again, these sorts of experiments in this animal and other animals. Eventually, hopefully, we'll have something very interesting that tells us about the role of co-transmission in the fly, which will help us understand and relate it to our information about co-transmission in the mouse. And then, hopefully, co-transmission in the human. So where can we go from here? Another place is bioenergetic demand. I said in the very beginning that you can have a co-transmitting neuron, and that this co-transmitting neuron, though it's more expensive than a monotransmitting neuron, it's going to be less metabolically expensive than two monotransmitting neurons. And so it would be interesting to know, given these proposed computational capacity increases, how much better exactly is co-transmission, metabolically cost speaking, than monotransmitting. It would be really interesting to get like a general understanding of how energy availability affects neural transmission under different morphological constraints. I'm very interested in sort of the trade-off between metabolic costs and function that is achieved, or the information processing that is achieved by the creature. And so this is a really cool place to go. Stretch goal would be a mathematical synthesis understanding of metabolic and variational free energy, where variational free energy has to do with understanding the information processing of whatever it is you're modeling, whereas metabolic just has to do with the cost of that information processing. And currently we have sort of two different maths for describing both, and I want to understand very deeply how these things relate. And so I'll be working on this as well in conjunction with everything else. Thank you.